I am Brooke Sadler, and this is my husband, Gabe Haller, and we are sharing our work on the contribution of rare variants and copy number variation on risk of sporadic CJD and pleiotropy with other neurodegenerative diseases. Okay, so before I begin, I'll just define a couple terms in case people are unfamiliar. So pleiotropy, from the title, just means when one SNP affects multiple disease phenotypes. And then heterozygosity is when you have one of each allele at a given position. So for instance, in the example of the M129V codon, if you have an M and a V, you are heterozygous at that position. Whereas homozygosity is when you have two of the same allele. So if you're MM or VV, you would be homozygous. So as some of you might know, sporadic CJD makes up the majority of all CJD cases at about 85%. However, unlike inherited CJD, there are a few known genetic risk factors, the most famous of which is the M129V codon in the PRNP gene. And it's been shown that heterozygosity at this locus is protective against sporadic CJD, while homozygosity for either allele confers increased risk. The genotype is also known to influence the clinical phenotype, for example, with the subtypes of sporadic CJD. And it's also been found that heterozygosity here is protective against variant CJD, so acquired CJD. Another important codon that is the E129K codon in PRNP as well, where heterozygosity has been shown to be protective against sporadic CJD in Asian populations. So recently, there was a Genomewide association study that discovered the first non-PRNP association with sporadic CJD. This was very exciting research done by Simon Mead and company. And they do find, as you see here, that there was an association with PRNP. However, they also find associations with two new genes, GAL3ST1 and STX6. This is I should have said, this is a Manhattan plot where each dot is a SNP, and then these are all the chromosomes. The line represents the cutoff for genome-wide significance. So for our project, our objectives were, number one, to do case control analysis of rare variants and copy number variants associated with sporadic CJD, using samples that we got from Dr. Brian Appleby at the National Prion Disease Surveillance Center, and 1,000 elderly cognitively normal controls that we have here at the university. And then aim two is to perform combined analyses of Alzheimer's disease, frontotemporal dementia, and Parkinson's disease with sporadic CJD to identify potential pleiotropic associated loci. As we know, there is some overlap in multiple forms of neurodegeneration. So we, COVID did sort of delay getting our samples, but we did manage to whole exome sequence 71 of our samples. And so those were from Brian Appleby. And we just got the data back recently. And we also have recently received 325 more samples that are also from the Prion Surveillance Center, but these were sent back to us from Simon Mead, Dr. Simon Mead, who was using them as well. And we are getting 1,000 cognitively normal control exomes very soon from our friend and collaborator here, Dr. Carlos Cruchaga, who studies Alzheimer's disease, and that's why he has these samples just around.
So hi, I'm Gabe Haller, husband to Brooke Sadler. So just to start, I'd like to explain a few terms that I'll use in the next few slides. So one is codon. So codon is a term used for 3 bases of sequence that codes for a specific amino acid within a protein. The term exome refers to the portion of the DNA within an individual that encodes the proteins. So it encodes the exons of those proteins, so the exome. The word exon, which I just used, is the portion of a gene that encodes the protein. So genes have introns and exons. The introns are spliced out of the RNA, and then the exons are spliced together and used to encode the protein. And CNV stands for copy number variant. And so that refers to a genetic variant that is a duplication, deletion, or inversion of a segment of DNA. Rather than a single nucleotide, it mutates a large portion of the genome. And so I'll detail a bit of the work that we've done on the 71 exomes that we've sequenced so far from sporadic CJD patients. So far, the first step, what we've done is perform exome sequencing on these patients, DNA samples. So in short, exome sequencing is the sequencing of the protein coding sequence within a person's genome. And so that's performed by taking the whole DNA from a patient and extracting by hybridization with RNA probes that are annealed to the DNA that correspond to the exons within the protein coding genes in the genome. So we take those DNAs, we perform this capturing protocol, and then we perform next generation sequencing on the DNA. We then align the reads that we obtain from the samples to the whole genome and perform genotype calling. And now what's important to note is that the majority of genetic variation in the general population and in all people, regardless of disease status, is extremely rare. So if we look at the relationship between the proportion of genetic variation here, single nucleotide polymorphisms, abbreviated SNPs, against the allele frequency, so how common that variant is in the population, what we see is the majority of variants are in this low range that are extremely rare in the general population. And so a lot of our techniques these days using these sequencing methods is to understand the role of these more rare genetic variants in disease susceptibility. And that's again sort of displayed here. When we compare the relative effect size, so how likely that variant is to contribute to disease against the allele frequency or the frequency of that variant in the population, and the color of these boxes is sort of the power to detect an association with that variant. And we can see that at very high frequency or very high effect size, so for instance, the variants in PRMP that caused the genetic forms of CJD would fall in this range here, where extremely rare in the population, but very much causative. They have extremely high effect size. And GWAS, so genome-wide association studies, sort of fall in this category where they're high frequency, but they only confer a very mild risk increase to patients. And sort of where we find ourselves in a lot of our genetic associations is in this middle range, where we are trying to identify somewhat rare variants that contribute a little or a lot to disease susceptibility, but aren't necessarily causative. So the first way, and Brooke alluded to this when she showed Dr. Mead's GWAS that was recently published, is to do a single variant analysis. So simply comparing the frequency of a genetic variant in cases and controls, where cases in our case are sporadic CJD patients and controls are patients or individuals from the general population. And so what we can do is simply run a Fisher's exact test or a chi-square test comparing the relative frequency of a given genetic polymorphism in those two groups and derive a p-value of the probability of that occurring by chance. And so if that's very low, then the probability is significant and likely suggests that the association is real and causative. And so we performed first a single variant association analysis with all of the genetic variants we obtained from exome sequencing of the 72 sporadic CGD patients and compared that to 2,230 in-house unrelated European American controls that we have. Unfortunately, we see no genome-wide significant association. So here on the left side, we have what's called a Manhattan plot, as Brooke alluded to earlier, is every single variant in the genome is a single dot, and its height on the y-axis is its significance. So the negative log of the p-value, so the lower the p-value, the higher the dot is on the y-axis. And the red line is genome-wide significant, so 10 to the minus 8, which is 0.05, a nominal p-value, divided by the number of variants that you've done. And so that's generally around 10 to the minus 8. And unfortunately, we see no SNPs associated at that level at this point. But we are, as Brooke said, sequencing another 325 sporadic CJD patients, and so hopefully we'll have more power in the near future. And then secondly, one of the things that we often do with exome sequencing is perform gene burden analysis. And instead of every single variant, we in fact sequence and combine rare variants across genes. So in this case, we take gene A, we take all of the rare variants and cases and collapse them and compare that number, here 8 out of 10, to controls. And we do that for each gene in the genome. And similarly, we can do that by grouping genes, where we take all of the variants from multiple genes that have related function and compare that frequency in cases to controls. And so we did that again with our 72 sporadic CGD sequenced patients and identified, although no genome-wide significant or exome-wide significant genes or gene terms, we found some interesting genes near the top that we hope may replicate in a larger data set. So these genes are PUM2, STK24, and interestingly, at the top of the gene set burden analysis, we find a gene ontology term that contains genes involved in the regulation of axon regeneration. So these are very promising preliminary results that we hope may replicate in a larger data set. So again, like the genes that we found, PUM2, for instance, was very interesting because it occurred in 6, rare variants occurred in 6 sporadic CGD cases and no controls out of 2,230 patients. And that was a very near, potentially causative, but we'll have to do a lot more work to understand that relationship. It encodes an RNA binding protein that has been shown to be a negative regulator of lifespan and mitochondrial homeostasis, and has been shown to have abnormal function and in various situations. And in general, RNA binding proteins have been shown to be the cause of multiple neuromuscular degenerative disorders. We've also found other interesting genes like STK24, which is serine threonine protein kinase. And we found 2 genes involved in the proteasome in which they have occurred only in sporadic CGD case and no controls, which we'll look into more in the near future. So Brooke will finish up, and thank you for your time.
Okay, it's me again. So another analysis that we did is looking at copy number variation. And to do that, we use this program called Fishing CNV that allows for copy number variation in exome data, and it is made specifically for rare variation. And so our preliminary results from this analysis oh, I should say 72 samples, sorry. We compared our samples to 213 unrelated controls here. And we found that 3 CJD cases had significant partial duplications of MTMR2, myotubularin-related protein 2. And homozygous loss of this causes Charcot-Marie Tooth disease type 4B1, which is a severe demyelinating neuropathy. And so here you can see we've just plotted the location of this duplication relative to the gene here. This is MTMR, so you can see it's only a small portion of this gene that's duplicated here. So there's a lot about deletions of this gene, right, that cause this demyelinating neuropathy, but little is known about the effects of duplications in this gene. So potentially it could be some kind of gain of function. Again, would need to look at more people to replicate this. We did find that 2 CJD patients had significant duplications on the X chromosome, which included this gene, HSD17B10. And this gene is interesting because the brains of individuals with Alzheimer's and of animals in an Alzheimer's mouse model exhibit abnormally elevated levels of this protein. And this overexpression disturbs the homeostasis of neurosteroid metabolism, disturbs the balance between pro-inflammatory and anti-inflammatory cytokines. So this increases neuroinflammation leading to deterioration of cognitive function. So again, needs more validation, but potentially interesting. So in conclusion, we have identified a number of intriguing candidate genes with an enrichment of rare protein-altering genetic variation in sporadic CJD. We've used gene burden and gene set burden analyses on our exome sequencing cohorts, and this is a powerful approach to find genes that could confer increased risk for sporadic CJD. Rare copy number variation is another avenue to look at, and we can identify these via exome sequencing, and these also may play a role in sporadic CJD risk. Additional sporadic CJD case exomes, so these additional 325 patients that we will sequence will greatly improve our power for all of these associations that we've discussed, and hopefully find novel genetic causes of sporadic CJD. So we would like to acknowledge our collaborators, Dr. Brian Appleby and Dr. Simon Mead. Thank you for working with us to get us the samples during this trying time. And our collaborators at Washington University in St. Louis, Dr. Carlos Cruchaga, who is giving us his cognitively normal control exomes. Christina Granette, she allowed the use of some control exomes that we did use in this data. Dr. Jorge De Paula is my mentor. Thank you to him. And Dr. Linda Pike has given advice and is also my mother. And we would like to thank our funding from the CJD Foundation, specifically the Jeffrey A. Smith Memorial Grant, the Tom Stevenson Memorial Grant, the CJD Foundation Grant, and the Strides for CJD Grant. And lastly, I want to thank my father. And all of this was for him and everybody else that we've lost to this disease. So thank you.